Seth Research Laboratories in California have recently demonstrated that in some tumors, cancer cells are killed by marijuana, while the other healthy cells are left untouched. Cells that stop moving and become still white dots are dead cancer cells. The ability of cannabinoids to kill bad cells while protecting healthy ones is particularly important when we're talking about brain cancer because of the so-called blood-brain barrier. The brain has to be sheltered from outside influences that might hitch a ride on the bloodstream and cause havoc. What is exciting and unique about cannabinoids is that they can pass through the blood-brain barrier because of their slippery, fat-loving nature. Cannabinoids get right into the brain's cancer cells by moving easily through the cell's membranes, which are also composed of lipids.
As the primary active constituent of the medicinal plant cannabis, also known as marijuana, most people are familiar with the euphoric effects of this compound. After entering the bloodstream, this compound becomes available to individual cells in the brain. Delta-9 THC interacts with specific cell surface receptors. It is analogous to a key entering a lock. This triggers a reaction inside the cell. Unexpectedly, this reaction in normal cells is significantly different compared to the reaction in cancer cells. In cancer cells, these reactions lead to cell death. In normal cells, it doesn't. This surprising difference is likely due to two things: a greater number of receptors on the outside of the cancer cells, and a different type of reaction inside the cancer cells as compared to the normal cells.
Intriguing answers started appearing in the early 90s when researchers pinpointed receptors in the brain and the body that bind with the cannabinoids. Receptors can be described as locks on the surface of a cell, and when the correct key binds with the correct lock or receptor, it opens the door and delivers messages. Sometimes the message is that the body is feeling pain. Other times the message may be that there's an invader and the immune system must attack.
Scientists located two receptors, cannabinoid receptors, one called the CB1 receptor, mainly in the brain, and the other is the CB2 receptor, which is mainly in cells of the immune system.
The CB1 receptors are extremely abundant in the brain, but they're also found all over the body in the major organs, the heart, the liver, kidneys, and pancreas. After finding all these locks that accepted the cannabis key, researchers made the next big discovery. The human body makes its own cannabinoids, called endocannabinoids.
We have this whole elaborate system where we have these receptors in our brain and in our immune system, and these circulating chemicals that we produce ourselves that really are very, very similar to the chemicals in the marijuana plant.
The only difference is that the endocannabinoids that we produce are in such small quantities, and they're also rapidly degraded, so that therefore we are not high all the time, or we don't have that feeling of euphoria all the time.
Dr. Prakash Nayagarkati is a professor of pathology and microbiology at the University of South Carolina. For the last decade, he's been doing research on what's become known as the human endocannabinoid system.
The precise functions of the endocannabinoid system is it's still being understood, actually.
The discovery of the system, however, is already revealing clues that have bolstered the personal stories of relief. The areas of the brain that control nausea and vomiting, chronic pain, and epileptic seizures all have cannabinoid receptors.
What do these things do? Well, in the brain and the nervous system, the cannabinoid system seems to exert kind of a dampening effect. It's kind of an internal, I like to think of it as a neurological shock absorber.
And when they looked in other animals, they found these receptors were present in basically all animal species. So why do dogs and monkeys, for example, need to have cannabinoid receptors?
They must be playing a very critical role in trying to maintain some of the physiological functions.
The whole development of the fact that there are cannabinoid receptors in endocannabinoids has really given drug companies and pharmaceutical investigators a lot of opportunity to try to manipulate the body's own cannabinoid system.
That's because now they can create synthetic drugs that target the receptors instead of taking chemicals from the plant. By avoiding the plant, they get around the controversies and complications of its Schedule I status. A search of the U.S. patent database reveals numerous large pharmaceutical companies have filed recent patents claiming their cannabinoid receptor drug has the potential to treat almost everything: multiple sclerosis, Alzheimer's, Parkinson's, rheumatoid arthritis, Tourette's, epilepsy, heart disease, obesity, various mental illnesses, and the holy grail of medicine, a cancer cure.
We feel that these cannabinoids give us an opportunity to study their functions, and see how we can exploit them, how we can manipulate these cannabinoids and the receptors to find cures for a large number of diseases currently in which there is particularly no cure.
As an immunologist, Dr. Nayager Katti and his researchers at the University of South Carolina are exploring the impact cannabinoids and the CB2 receptors have on the human immune system. Both U.S. reports on marijuana found the cannabinoids do suppress the immune system, and previously this was seen as a concern. But Dr. Nayager Katti believes tamping down the immune system could be a good thing. Is a concern. But Dr. Nayager Katti believes tamping down the immune system could be a good thing.
There are about 80 different autoimmune diseases, and basically autoimmune diseases are triggered by your immune system going haywire, getting hyperactivated and destroying certain cells and certain tissues.
In multiple sclerosis, for example, the immune system suddenly begins attacking the brain and the spinal cord. Rheumatoid arthritis and lupus are similar diseases caused by the chronic inflammation associated with an immune system gone amok.
The cannabinoids dampen down response to bacteria or foreign agents or injury to tissues.
A lot of scientists and researchers are interested in those properties of the cannabinoids, in trying to see how we can suppress the immune response in autoimmune disease conditions.
For Dr. Nayager Katti, the research has gone far beyond treating inflammatory problems. He's narrowing in on the potential of cannabinoids to kill immune cells that have mutated and become cancerous. Once they become cancerous, they no longer die a normal cell death. Instead, they begin growing and spreading uncontrollably.
We were one of the first labs to demonstrate basically that not only the immune cells, the normal immune cells express these cannabinoid receptors called the CB2 receptors, but also that when these immune cells get transformed and they become cancer, to our surprise we found that these cancer cells continue to express these CB2 receptors.
This was an exciting discovery because the CB2 receptor can act like a target for the cannabinoids. Once they bind with the receptor, they can tell the cancer cell to die.
So basically telling the cells basically to commit suicide, and that's what they do. And we demonstrated that that would be the mechanism by which the cannabinoids can kill the cancer, and therefore it can be used effectively as an anti-cancer agent.
Dr. Nayager Katti and his researchers were able to eradicate almost 100% of the cancer in test tubes. But they were skeptical they would see similar results when they moved on to tumors in mice.
To our surprise, we found that almost 25 to 30% of the mice completely rejected the tumor. They were completely cured. And in addition, we found that the remaining mice also, there was a significant reduction in the volume or the size of the tumors as well.
The lab results have been so promising that Dr. Nayager Katti is beginning clinical trials with leukemia patients. There's no doubt in his mind that the cannabinoids, either from the plant or lab created, will play a major role in medicine in the future.